Good morning, good afternoon, or good evening, and welcome to the latest Novogene webinar, wherever you are in the world. Today, we dive into the world of long-read sequencing, a breakthrough technology that has reshaped genomic research. Our latest webinar, Long-Read Sequencing, Expanding Horizons in Genomic Research, will explore the transformative potential of long-read sequencing and other insights into applications and advantages. As many of you know, we've been running our webinars for a while now. So if you'd like to explore other aspects of next generation sequencing, all our previous webinars are available to view for free on our website. This includes our ever popular beginner's guides webinars. We'll send you a link to our website in our follow-up email. A little bit of housekeeping before we start. Presentation will run for around 45 minutes with 10 minutes for questions at the end. Please submit your questions via the Q&A button at the bottom of your screen. You can upvote a question by clicking the thumbs up symbol next to it. We'll try and prioritize the most popular questions. If we don't have time to answer your question, we will come back to you via email after the webinar. Please don't use the raise hand functionality. Just send any questions via the Q&A or message us directly if you have a technical issue. We'll share the slide deck and a recording of the webinar via email. You can expect this on Friday. If you are studying and need a certificate of attendance, there'll be a link in the follow-up email that will display a certificate you can print directly from your browser. Finally, there'll be a short survey that pops up when the webinar finishes. Please take a couple of minutes to complete this as it helps us to plan future webinar topics. I would now like to introduce your host for the webinar, our account manager for Berlin Biotech, Brandenburg and Saxony, Julia Camarelli. Julia, over to you.
Thank you, John, and hi, everyone. Thanks for joining our webinar today. So as John said, my name is Julia. I'm an account manager for Novogene in Germany, and I'm your host for today. So in a minute, I'll pass over to Dr. Shichi An from Novogene, who will discuss the principles of long-read sequencing and the technology's impact on current genomic research and the great potential it holds for this field in the future. So before I hand over to Dr. An today, I just want to say a few words of introduction about Novogene, our company, just for the benefit of anyone who is joining us for the first time. So let's go. So Novogene was founded in 2011, and we've been growing rapidly since then. Our company mission is to advance genomics and improve life. And we strive towards this by offering services covering a huge range of applications, all with real-life implications, from complex disease research and oncology to agriculture and conservation to food and water safety. Our European chapter is based in Cambridge in the UK, and our lab there is ISO 17025 and ISO 15189 certified. And this has enabled us to expand the range of our clinical sequencing services as well. It is a state-of-the-art lab. It's equipped with Illumina NovaSeq 6000 sequencing machines, and of course with the latest NovaSeq X Plus system. So we've always provided fast turnaround times as well as high quality data, but this is now better than ever thanks to the Falcon 2. This is our intelligent sequencing platform that has increased our monthly capacity by 200% compared to manual steps. So we're very proud of this. We've also have our supercomputing cluster, which lets us offer bioinformatics services and store your data in the UK. However, at heart, we are a global company. We offer services across Asia and the US, and of course, Europe. The Falcon 2 is installed across several of our labs, so our services are standardized globally. And the same goes for the NovaSeq X Plus system, which keeps us at the forefront of the latest in genomic technology. We have over 2,500 employees, and we serve over 5,500 customers in world-class universities, biotech companies, hospitals, and so on. So we can provide sequencing for even the smallest and the largest projects. We provide a full range of next-generation sequencing, just to mention a few: whole genome sequencing, whole exome sequencing, various types of RNA sequencing, metagenomic services, epigenomic services, the list goes on. We also offer pre-made library options for most of these services, and our excellent bioinformaticians can provide options for analysis in most cases as well. Our customers use our services for a wide range of research topics, from cancer and rare disease research, microbial cell factories and gut health, to agriculture and animal health. Many other companies offer these services too. We know actually the NGS market is pretty crowded. So why Novogene, right? Well, we believe there are a lot of good reasons why you should trust us with your project, and why actually so many brilliant researchers and clinicians already do. As I mentioned, we have the ISO 15189 certification, which gives you peace of mind that even clinical samples are in safe hands. We have our intelligent NGS delivery platform, the Falcon 2 that I mentioned. It has really leveled up our capacity, and it standardized our processes globally, which resulted in our fastest turnaround times and highest data quality to date. So we are very proud of this, because it just really highlights the innovation that we strive for here at Novogene. We really work hard to keep pushing the frontiers of genomic research. We have the latest in sequencing technology installed across our network of labs. Most recently as well, the PacBio Revio system, which is relevant for our long read discussion today, and of course the NovaSeq X Plus system, which allows us to continue offering our customers best-in-class service. We know that the benefits of our in-house innovation are proven with our record in the scientific community, which we are also very proud of. We've been cited in over 790 research papers with a total impact factor of more than 6,120, and we have over 37 NGS-related patents. Our technology is helping to make the NGS process more efficient and effective, all the while improving the quality of the data that we deliver. So our community is growing all the time. Our expert European sales team speaks over 10 languages, and we're located across Europe. We're ready to assist you in your next project whenever you are. So even if you don't have samples ready to send at this point, we can still benefit from our educational outreach like today's webinar. And we can also put you in touch with your local representative to learn how NGS can work for your upcoming projects or future plans. We regularly host seminars as well in universities and institutes across Europe, because we want to educate the research community about the applications of NGS. So if you would like us to organize a seminar near you, please feel free to get in touch. You can also keep an eye out on our social media for the announcement of dates already organized in your city, and you can email us whenever you need. Aside from seminars, we offer a lot of educational resources, from webinars like today's one to blogs and even case studies. And you can find all of this info on our website. So just to briefly introduce you to our wonderful European sales team, as you can see, we're based across the continent, and we're all super happy to help you with your sequencing projects. So if you would like to speak to your local representative, you can email us, as I said, at info@novogene-europe.com, and you'll hear back from us very quickly. So that's it for my introduction. Thank you so much. And I'm now happy to hand over to Dr. Shichi An, who will talk to us about long reads.
Okay. Thank you very much for the introduction. And could you please stop sharing so then I can set up my slide? Okay, yeah, Julia. And hello, Julia. Okay, is still there? Could you please stop sharing, and then so I can set up the slide. Hello. Hello. Oh, thank you. You're still sharing.
I should say, I think if you try sharing now, if I stop sharing, there we go.
Okay, thank you. Perfect. Yeah, just bear with me and I'm just going to set up my my pointer up just for a sec. Okay. So with quite a bit of information we're going to cover today. So without further ado, I'm just going to give you a quick overview about what we're going to talk through today. We'll start with an introduction about the evolution of sequencing tool, and then we'll spend some time.
Sorry, you're on your you need to swap your notes around.
Oh, okay. Sorry.
Presentation.
Yeah. Okay. Here, display, and I'm going to start. Okay. Okay. Sorry.
Perfect.
Okay. And then we're going to move on to some discussion about the sequencing platforms. This includes PacBio and Oxford Nanopore platforms. And then we move on to the important key things to make sure we have a successful long-read sequencing project at the high molecular weight of DNA extraction. And then we'll spend the rest of the time to discuss applications and that can be applied using long-read technology. And in the end, we're going to have a quick summary to recap what we talked about today. Okay. So we start with a little bit of introduction of the sequencing tools evolution. So I think everyone here probably know the first generation sequencing, with the Sanger sequencing. So this is able to sequence a short fragment that is ranged from 500 BP to 1,000 BP fragment. This is relatively slow and expensive due to its low throughput capacity. And then the second generation sequencer is also known as the high throughput sequencing platforms. And this is the generation that revolutionized the sequencing field, basically. And the most broadly used platforms to date is still Illumina platforms. So they featuring a high throughput sequencing capacity, but they're still considering as short read platforms. They're able to sequencing about 50 to 500 BP fragments. They're faster and more affordable. And more recently, there's the development of the third generation sequencing, and the most well-known platforms are Oxford Nanopore platforms and also PacBio. So both of the platforms are able to sequence native DNA in real time with a single molecule resolution, and both of them are able to sequencing tens of kbs fragment on average. So they're featuring the long read sequencing, the real long read sequencing. So for some of you may not know that long read sequencing was also selected as the method of the year for 2022 by Nature method. Okay, here I'm just going to borrow the title from the publication that long read sequencing powers a more complete reading of a genomic information. So help us to read the book of life even better. Okay. So now we're going to spend a little time to discuss about technologies actually behind those platforms. So at Novogene, we offer services on both PacBio platforms and Nanopore. Okay, so for PacBio, we offer services on both PacBio Seco2 and the 2E platforms, and also the newest PacBio platform, Rabio. So we are a certified service provider. And for Nanopore platforms, we offer services on Oxford Nanopore Promethean, which is the production scale sequencer. And then we're also certified service provider there as well. Okay, now I'm going to start with PacBio sequencing technology. So this is known as single molecule real-time sequencing, so smart sequencing. So this is how the smart cell looks like in real time. And this is looking from the bottom of the smart cell. So there you are, a size of a fingernail in general. Okay? And then each of the smart cell contains millions of zero-mode waveguides, as you can see the wells here. So they are called ZMWs. And then after library preparation, it's going to generate a SmartBell library templates. They are circular DNA molecules. Okay, we're going to come back to this point of later. Okay. And then a single molecule of the DNA, those templates then can be immobilized in each of the ZMWs. As you can see, the seeds immobilized in those wells here. Okay. And after that, anchored polymerase then will incorporate a fluorescent labeled ATCGs basically during the elongation, and then based on which color was fluorescent emitted when the base is incorporated, then we will know what was the sequence of the original DNA. Basically. Also, because the technology is basically measuring the nucleotide incorporation kinetics in real time, and there is a chance that it's able to direct measuring the modification of DNA as well. So I'm going to come back to this point a little bit later in the talk. So here I wanted to come back to the circular shape of the templates. So this is actually enabled technology able to sequence the same molecule multiple times. Okay. So this actually leads to the different sequencing mode. Here I'm going to talk about for PacBio. And it had two different sequence modes. So one of them is known as continuously long read CRR mode. Another one is known as the circular consent sequence mode, CCS. So what's the difference between the two? So for the CRR mode, this will allow the template to have a larger insert, but the polymerase is only going to be able to go through one pass of each molecule that is sequenced. Okay. An advantage of this is that it allows to sequence a larger insert, but a disadvantage is that it still has a quite high error rate under CRR mode. However, for CCS mode, so it only allows to sequence the smaller inserts, but actually the polymerase will be able to go through the inserts multiple times. That's generate multiple subreads of the same region. Because error is at random, then by extract consensus reads sequence from the multiple subreads, then you're going to end up with a high fidelity reads, also known as high-fi reads. So really, really high accuracy. So this will requires the polymerase goes through the insert at least 2 times in order to generate high-fi reads. Okay. And then the mentioned we have services on different platforms, and especially the newly launched Revio system. And here is a quick comparison between the two different platforms, and I'm just going to highlight some of the major differences. So the first thing is the supported smart cells are different. For SQL2 and the 2E systems only support 8A smart cells. 8A basically means it contains 8 million ZMWs. And for Revio, it contains 25 million. So that means the capacity significantly increased here. And for running mode, SQL2 and SQL2E are able to run both the legacy mode, CRR, and also the CCS hi-fi mode. However, for Revio, this is really optimized to just run for CCS mode to generate a hi-fi read. Okay, so for hi-fi data yield, obviously, the Revio has significantly increased on the yield. Okay, so please note here, and those numbers, the hi-fi data yield really depends on the sample quality and also the insert size as well. For the SQL2 and SQL2, it has a little bit more flexible running time because it's running for both different modes. But for Revio, this is optimized for the hi-fi, so the running time is fixed. And then another key improvement of the new system is that it's able to run 4 smart cells independently in parallel. So that also helps increase capacity of the equipment as well. So methylation, I'm going to just briefly mention here that for the CRR mode, it's able to detect 5MC, 4MC, and 6MA. And then for hi-fi mode, for both of them, they're able to detect 5MC at CPG sites. I'm going to come back to methylation a little bit later during the talk. Okay. And then let's move on to Oxford nanopore technology and how nanopore works. So basically, nanopore is protein pores that forms on membrane, and the pore is nanometers in size, and that's why it's called nanopore. And so how this generally works is that one of the protein is going to unzip the double helix, and that allows one of the chain, one of the string that's passing through the pore. Okay, so when the base passing through the pore, it's going to cause a disruption of the ion current within the pore. And by reading this disruption, the change of the current, then we're able to decipher what was the base is passing. But this is basically how nanopore works. And then the most recent advancement for nanopore technology is they launched the Q20 plus chemistry. Okay. So this includes a new pore as R10.4.1. So the pore is improved design for tuned enzyme and faster speed and tighter speed distribution, and also introduced a new motor E8.2. So this motor has improved movement property with some more consistent movement as well. And so combining those two, so basically it's now able to generate higher accuracy readings. For single molecules, able to reach accuracy of 99% or higher. That's why it's called the Q20 plus. This includes the V14 kit chemistry here, that's including the pore and also the motor, okay, E8.2. And then at the moment, Novogene now is able to provide a service with the Q20 plus updates for all our European customers as well for DNA sequencing. Now we're going to move on to the starting material, which is key to the successful long-read sequencing project, is the high molecular weight DNA extraction. So if you want to have a good project, a successful project, it should really start with a high quality, high molecular weight of DNA. Okay. So when we talk about high quality, what we should considering, okay? And those are the four things that we should consider. The first is quantification, basically concentration of your DNA. So this is usually measured by qubit fluorometer. So this is a fluorometric based equipment. So it is less affected by the presence of contaminants. Therefore, it offers better, more accurate concentration reading. Then high quality also means the purity of the sample needs to be high. And this is usually measured by nanodrop spectrophotometer. So this is good for measuring a DNA purity, but it frequently overestimated concentration. This is not so ideal for accurately reading a concentration. But for high molecular weight DNA long read project, we actually we need to actually read the concentration on both equipment, and then the reading between them should be less than 50%. This is very important. If the difference is too big, that means the quality of the DNA is not good enough. And then also size is important to measure as well. So this is usually measured by post-field gel electrophoresis and PFGE. So this is used to approximate the size distribution of your DNA sample. The high molecular weight DNA does not migrate well or separate well on the traditional gel. So this is really needed to be more accurately estimated the size of the sample. So another important thing to consider is actually trying to minimize DNA damage. So to considering to inactivate nuclease, maybe considering to use a tissue and a nucleic acid stabilizers and avoid phenylchromophore, and also avoid hydrolysis of anglexyl bonds. That's why using fresh sample is very, very important as well. So the key takeaway message from this slide is always aim to get molecules as long as you can, as pure as you can, and also as fresh as you can. So the key thing here probably should bear in mind is that you can't really sequence in what you don't have. So high quality DNA is really, really important. Okay. So here are some general tips and best practices should consider when you prepare your DNA extraction. So always use fresh or flash frozen tissues, and the flash frozen tissues just store at -80, and importantly avoid freeze thaw cycles. For blood sample, they should not be stored at 2 to 7 degrees for too long. And considering inactivated nucleic acids and also DNA binding proteins. and remove all RNAs, and also avoid chemotropic agents because this causes damages as well, and avoid an oxidative agent, as I mentioned, avoid phenol and chrome-4. Okay. For suspend or elute DNA, I use a low salt buffer. Okay. As I already mentioned, check concentration on both nanodrop and the qubit, and then the differences between them should be less than 50%. And also importantly, ensure DNA is pure. Okay. Well, during the preparation, okay, when you're handling the DNA, you should avoid vortexing and using wide board tips. And importantly, if you use a kit to prep your sample, always adhere to the kit manufacturer's guide closely. And so here I'm going to share some good news, and that recently Novogene collaborated with Echo Life Sciences company with an expert in nucleotide acid extraction. And then we're now able to offer high molecular weight DNA extraction services to our European customers. Okay. So please note that all the projects requirements will be evaluated to make sure the optimal outcomes. Okay. If you're interested in, please feel free to reach out. Okay. So now for the rest of the talk, I'm going to focus on different applications of long-read sequencing. Okay. And so the first application we're going to introduce is whole genome sequencing, de novo assembly. I think for most of the people, when you think about long-read sequencing, this is the first application considered. So the major advantages long-read sequencing brought in here is that it offers a simplified and less ambiguous genome assembly. So this is like you putting a jigsaw together. Okay, for long-read, you're actually using larger pieces. So you can see it's going to make the sample much easier compared to using short read, smaller pieces jigsaw. So it helps to make it much simpler. You need less reads, and also the relation between different reads are much easier to know as well. So less ambiguous assembly. Okay. So here I'm just going to briefly introduce some of the concept different level of assembly here as well. So people usually mention. So for assembly, it starts with reads. Basically, they are DNA fragments, and then overlapped and oriented reads then can be assembled into contigs, and then jointed and oriented contigs then can be assembled into scaffolds, and jointed and oriented scaffolds then can be assembled into chromosome. Okay, and then those scaffolds actually can be further arranged if you take into account of the interactions between contiguous and genomic loci. Okay, so this actually will require data from HiC-seq. Okay, so combine those information, then this will be able to generate a chromosome scale scaffold. Okay. So another new concept, recently more brought up by a lot of researchers, is telomere to telomere T2T assembly. So this is actually aimed to generate a gapless or near gapless DNA sequencing of the entire chromosome. So obviously long-read sequencing can actually help assembly at different level. And then the other important thing to consider is the quality of the sampling. So here's some general perspectives you should consider when you assess the quality of the assembly. And so this include contiguity, completeness, and correctness. So contiguity means that ideally you should have fewer sequences, but longer sequences to cover your assembly. So N15 is usually used as a parameter here to judge this. So N15 is the length cutoff for the longest contigs that contain 50% of the total genome length. And so for long read assembly, N50 reaches 1 MB. That usually means it's a very good assembly. Okay, so from completeness, this is very, very important as well. Basically, it assesses the assembly size, is that correct or not? If all the core genes are identified, and also the read mapping and assembly coverage as well. So one of the parameters that is usually used, or the score that is used to assess completeness, is called BISCO. score. So this is actually looking for the presence or absence of highly conserved genes in an assembly. So generally speaking, when the viscose score is above 95%, that usually means it's a really good assembly. Okay? And also correctness is very important. Basically, it means all the index needs assembled correct, and also all the complicated regions, repeats, joints are arranged correctly as well. Okay. Now we're going to move on to the next application. It's still whole genome sequencing, but this time it's a resequencing. So basically, you have a reference genome there to map to. Okay. And then the advantage of long-read sequencing is actually it's able to provide a more comprehensive variant detection. Okay. Here you can see here for short read sequencing, when it comes to mapping, usually there's some common problems here because it's easily to have missing data that will leave gaps in the genome coverage that usually will limits the variant detection. Actually, for long read sequencing, this only requires fewer SNPs to make up the length of the whole genome. Okay, and also it's more easily to map on how one region of the genome is connected together. Okay. Importantly, it also has the ability to phase or determine which copy of a gene it is a maternal or paternal mutation that actually occurs in. So here's an example here. So basically, we have a maternal chromosome and the paternal chromosome. For unphased variant coding, all the variations is going to collapse into one mixed sequence. Okay, but for phased variant coding, each haplotype can be assembled separately. So that means they are able to know the mutations actually occurred in the maternal or paternal chromosome. Okay, this is going to provide a more complete picture of genetic variation. So here's a summarized slide that's to show that the long read actually able to span more variants type and provide more comprehensive detection as well. So start from a one base single nucleotide variants, and also small indels, usually they are less than 50 BP variants, but also importantly, very good at coding large structure variants. Okay, so here basically also demonstrate as able to cover more types of variants. Okay. So here are also some generalized recommendations for the coverage that's required for a whole genome sequencing with long-read technology. For gap filling and scaffolding usually requires more than 10 times coverage. The largest structural variance detection, again, usually requires more than 10 times coverage. For germline and frequent variant calling, 20 to 50 times coverage is recommended. And for de novo sample, that usually requires 50 to 100 times coverage. Okay, so please bear in mind this is generalized recommendation, and the coverage that is required really depends on the research aims and also the technology you choose for your study as well. Okay. Now let's move on to the next application as pan-genome sequencing. So this is actually looking beyond the single reference genome. So what is pan-genome? A pan-genome is basically a total collection of genes that share, okay, core genes, basically shared by a species, okay, include both common or the genes that is actually unique to individuals as well. And so by sequencing pan-genome, you're able to capture the full range of genetic diversity. So why this is important? So this information actually can provide insight into comprehensive diversity, adaptive strategies, and also pathogenicity information as well, and help us with precision agriculture. And so here's a very interesting example. So recently, this publication that showed researchers studied the maize pan-genome, and they actually have found that there's more genetic diversity within maize species and then between human and chimpanzees. Okay. And so how to generate a pan-genome? The key things here is that to generate equivalent quality genomes for fine samples with little variability, and then assembly needs to be more robust and accurate. So that's why the long-read sequencing comes into very helpful here to generate a good assembly, a quality assembly for all individual genomes, and then this will actually enable the analysis of the pan-genome. for functional and translational analysis. And then I'd like to also mention another application that has targeted long-read sequencing. So here I'm going to use the whole exome sequencing as an example. So this is based on Twist exome 2.0 panel and also PacBio hi-fi read technology. And so by combining those two technology, it's able to actually generate a robust coverage of difficult to sequence or difficult to map regions. So here is an example. So basically, this is the mapping from the hi-fi targeted enrichment sequencing, and then this is the short read data. Okay, so we can see this region here, the short read cannot map here, so it's unmapped, or there's only a few reads there. But for long read sequencing, and this region is actually well covered. And also this provides a high accuracy at variant calling as well. So here's some data from Novogene internally. And so you can see here, 16 times long read whole exome sequencing is already have much better variant calling compared to 30 times short read whole exome sequencing. And also, as I mentioned, it's able to generate and compute haplotype resolution as well through phasing. And also, and this can be quite cost effective. And if you wanted to identify rare mutations, and also there's a potential to actually apply to customize the panel as well. Now, I'm going to move on to epigenomic sequencing. So I briefly mentioned that both of the platforms can actually detect DNA modification during sequencing directly. Okay, so here's just some details here. And so for PacBio, because it's actually measuring the nucleotide incorporation kinetics directly during the sequencing, so the modification, for example, 5MC is going to impact on this kinetics, and therefore this information can be used to identify the modification. And there's no special library prep required, which means that you don't have to do a bisulfite conversion, for example. Okay, so for the coverage that is generally required for 5MC coding and CPG, that requires 30 times coverage, and for microbial methylation, so this can be called under CLR mode for 4MC and 6MA as well. But please be aware that in order to reach a high resolution and calling, that requires more than 100 times coverage. For nanopore, also the modification actually causes a different impact on the current when the base is passing the pore. Therefore, this information can also be used to directly identification of the modified DNA bases. Okay. And so for the coverage, this really depends on your study aim and also the complexity of the methylome being investigated. So you should also consider the level of the resolution that is required and combine all this information to determine appropriate coverage for your study and nanopore platform. Okay. So here, before I move on to an RNA applications, I just want to show you an example that demonstrate how long read can help with your research. So here is a paper that is published on microbial genomics late last year. So this is sequenced with Novogene. So here they had data generated from PacBio sequencing. This is generated from this study, but also they have data generated from Illumina and Oxford Nanopore from previous study as well. So here they did PacBio assembly and also did a gene and prediction and the functional annotation, and then they compared all the available assemblies, performed a comparative genomics analysis. So basically, this is a study that is looking at the global pangenome of the wheat fungal pathogen here. So I won't go into too much details. I just wanted to emphasize some of the key important things that are related to long-read sequencing. And if you're interested in the paper, please feel free to read the whole publication afterwards. So what they have found that they have identified this particular fungal pathogen genome. It has a lower divergence compared to other wheat pathogens. And importantly, using the PacBio data, they're able to pinpoint some effector transfer region. So that is actually only exclusive to those effector producing races. So this is very accurate. So the whole paper basically generated information to help us to understand the fungal evolution and help in development of targeted strategy for managing wheat disease. Okay, so a very interesting paper. And then for the next bit, I'm going to move on to RNA applications. Okay. And so first, I'm just use this diagram basically to demonstrate the advantages of using the long-read technology in RNA-seq. So here, for example, this is the gene, okay, here. And then course of alternative splicing, the potentially can generate two different types of transcripts. So basically, this is X and this is Y. So this is black and this is a red transcript Y. So for short read sequencing, when you want to identify isoforms, for example, for regions here, this is generates ambiguity here, because if you have reads obtained on those regions, you wouldn't be able to differentiate that it actually comes from transcripts X or transcripts Y. So you won't know. But actually for the long read sequencing, you're able to obtain the full length of the transcript. So that means there will be no ambiguity for isoform identification. This is one of the major advantages of using a long-read sequencing for RNA-seq to actually accurately identify isoforms. So here I just listed some of the mechanisms that can generate isoforms. For example, alternative transcription start site and also alternative transcription termination site. So long-read RNA-seq, then we are able to allow you to see the alternative start and end site. And also the isoforms can be generated through alternative exon splicing or intra-retention, and also the long-read RNA-seq or full-length transcript sequencing allows you to characterize splicing events as well. But actually, the application can go beyond just isoform identification. So here, just some examples here. So the first thing is a full-length transcriptal sequencing also can help to identify fusion transcripts. So fusion cannot just happen at gene level, but also happens at transcription level. Okay? So the long-read RNA-seq can help discover novel fusion transcripts, especially in clinically relevant genes to help us understand disease mechanisms, for example. And also the data generated from a long-read RNA-seq can help us discover non-coding RNA as well. And so to provide a more comprehensive view of transcriptional and translational regulation. Okay. So importantly, the data generated from long-read RNA-seq can also help us to improve genome annotation. So through the transcriptal information, this can actually help us to identify genes that may be missing in assembly to actually help us with to improve the genome assembly and the gene annotation through RNA-seq data. So this is actually a very important application for RNA-seq. Okay. So here I'm just going to quickly use another example to show you how this can help you with your research. So this is another paper that is published 2021 on Crop Science Society of America. This is also sequenced with Novogene. For this paper, the researchers built a reference transcriptome for a type of turf grass. Okay. So what they actually used here is the data from Illumina sequencing and also PacBio full-length transcript sequencing, also known as isoform sequencing. Okay. So what they did here is they did PacBio assembly for the transcriptome and also Illumina de novo assembly, and also used a combined assembly approach and joined both of the information together. Again, I just want to highlight some of the results. So basically, they developed a reference transcriptome for this turf grass species. And importantly, they showed PacBio isoform sequencing significantly helped to improve the short read de novo assembly. So the combined approach gives the best results, basically. And also, importantly, through the sequencing data, they're able to identify almost 8,000 transcripts that are long coding RNAs. So it's a very powerful tool, as you can see here. And for the last bit of the application part, I'm going to focus on applications in metagenome sequencing with long read. So generally speaking, it has three major advantages using long read sequencing for microbiome researchers. So first thing is it's able to provide a more comprehensive 16S, 18S, and ITS profiling. But this is using a full-length amplicon sequencing. Okay. This helps to reveal the true diversity and complexity of the microbiomes, and offers highly accurate to species and even to strain level resolution, which cannot be reached by short amplicon sequencing. Now, also for a long-read shotgun metagenome assembly, this can really enhance of the assembly as well to help to retrieve abundant high-quality metagenome assembled genomes, high-quality max. This also include a circular single contig max as well. Okay? And also, of course, the information generated from shotgun-based metagenome long-read sequencing can also be used for taxonomic profiling. Okay, so for today, I'm going to focus on to talk about the first two advantages. So I think for any of you, if you have studied microbiome and used an amplicon sequencing, you're probably very familiar with the short read applications. So different variation regions, considering what is the best regions to go for, what's the best primers to select. Okay, so this is very broadly used. But actually, there's a problem. This is very well demonstrated by the human microbiome projects. They've actually showed different amplicons all have different biases in texts they can identify. Okay, so here as the image figure that can very clearly show this. So the color red here means the claims that cannot be identified to the species level. It's different regions. You can see all of them. The more red they go, the less is identified. Okay, so you can see all of the regions, they're not perfect, they all have bias. Okay? Then when you actually use the full length, actually almost all of the claims then can be identified to the species level. Can see a sequence of the full length of the 16S genes provides a real and significant advantages over just sequencing a short region of the 16S genes. Okay. So here I'm just going to use another example to quickly show what can be achieved here. So this is published on 2021 on Frontiers in Microbiology, and this is a sequence that actually analyzed with Novogene and also use our Novogene platform for the analysis of the amplicon data. So here the customer actually used the PacBio-based full-length 16S amplicon sequencing and also ITS, full-length ITS amplicon sequencing as well. Here I'm just going to highlight some of the key points that showed in the results. Okay, so basically the 16S data is used to study the bacterial community, and then the ITS data is used to study the fungal community. And for both of them, you can see here, the analysis is able to reach a species resolution for both the bacterial community and also for the fungal community. Both of them is able to reach species level, which cannot be really reached by a short amplicon sequencing. So this is the key advantage I want to mention here. And then for the last application I wanted to mention is a long read, a shotgun-based metagenome assembly. So I already mentioned that this is really to help to recover more high-quality max through the sequencing. Basically, this can help to resolve complete genomes and plasmid directly from the community sequencing and correctly identify more max from the data, and can able to capture both a taxonomy and functional insights into the microbiome simultaneously, identify species, antibiotic resistant genes, and virulence factors And important that there's no DNA application involved in this process. Therefore, it's less bias generated as well. So very powerful tool. Okay, and then the last example I'm going to give you here is that a paper that is recently published, actually this year, on BMC microbiome, and then this is also sequenced and analyzed with Novogene. And so in this paper, they actually sequenced 242 horses and gut samples. Okay. And then they used Illumina sequencing and Oxford nanopore sequencing here as well. So for analysis, the researchers did a metagenome assembly, and then gene catalog construction, taxonomy annotation, and abundance profiling, and also metagenomic assignment. Okay. So here, just wanted to demonstrate what they actually achieved. Through the data, they actually assembled more than 4,000 max in the horse gut. So that is a very powerful tool. So over 4,000 max directly assembled from the data. And this represented an exhaustive microbial genome catalog for the horse gut microbiome, and provided a valuable resource for the discovery of performance-enhancing microbiomes and study of the horse gut microbiome. Okay. So very, very powerful tool in the microbiome research field. Okay. So in the end, I'm just going to quickly recap what we've touched today. So I hope I've convinced you that long-read sequencing using Oxford nanopore technologies and biotechnologies, they actually have improved resolution of complex genome regions and structural variants and detections. And also long-read sequencing is important in reveal accurate genome structures, transcript isoforms, and also epigenetic modifications as well. And also for metagenomic analysis that can also benefit from improved assembly of diverse microbial community. Another thing that I would like to highlight is that you can see from a lot of publications and the cases I showed today, they use a hybrid approach, basically take advantages of a short read and a long read technologies to enhance accuracy and completeness. And this is going to be the trend, I believe, in the future. And also, I hope I convinced you that long-read sequencing continues going to shape our understanding in biology and disease and in the intricacy of genomes. Okay, and I think that's all the main presentation for today. And if you have any questions, I'm happy to take some.
Thank you so much, Dr. Shichi. So yes, we do have a lot of questions actually. So we will get through as many as we can in the time we have left. So let's get started. So Sarah asked, do we have the service of long-read sequencing using Illumina platform?
No. So yes, you're right. Illumina also launched a computer read. So there's something you probably should aware that both PacBio and Nanopore, they are considered real long read because they are actually directly measuring the molecule that you wanted to sequencing. So Illumina is still, the fundamentals is still short read based. It's not a real long read. So here's a tricky bit here to bear in mind. So for example, if you wanted to identify methylation directly, Illumina's long read won't be possible, okay, for example. So yeah, there is some fundamental difference between the technologies.
Thank you for that. And next question is from Agnes. She asks, what is the maximum library fragment size that you can provide to the Revio system to obtain hi-fi reads?
Okay, so I think I briefly mentioned here, this is a good slide to say, because this, let me show you here. The Revio platform is really optimized for hi-fi. And for hi-fi, you need to have relatively smaller inserts and then to go multiple round. Okay, so generally speaking, that will be around 15 kb, and then the runtime is actually fixed as well, which means it won't allow you to go any longer. Okay, so 15 kb around that, it is the sweet spot for Revio, also for hi-fi sequencing by PacBio technology.
Thank you. Next from Samuel, he comments, PFGE is long and tedious to perform. So is it absolutely required for a project?
Again, this is some good news here. If you can't perform that yourself, actually, Novogene offers that service for you if you provide us with high molecular weight DNA. And so I hope that's a relief for you. Yeah.
Perfect. And next from Ramin, he asks, how does PacBio handle a high error rate of 15% on CLR mode using large inserts in terms of fidelity?
Okay, so it's a good question. Basically, that actually limited in a way is application. Okay, so this is really good if you want then to have longer fragments to do assembly, then you can actually use a short read data to polish the read or to support with the accuracy. So that's, it has a different application this way. Okay. So the error rate is still there, but the key thing is actually to look at the large size fragments, and then you can use the short read in a way to correct or to improve the accuracy. So it's still useful.
Great, thank you. And next question from Michael. He asks, do PacBio hi-fi sequencing runs include methylation calls as a standard? And how can this impact haplotype phasing, if it impacts it at all?
No, it doesn't impact with each other. Yeah, because it's just automatically getting the methylation information, and then and also the modification in principle can also be phased as well.
Great, thanks. And a question from Eslam. When we have a reference genome, is it feasible to perform genome resequencing for variant detection using long read sequencing, which is expensive, keeping in mind that short read would be easily aligned or assembled based on the reference genome and provide almost the same results?
It depends what you would like to do. So it's actually we see a trend. More people start to actually use long reads to do resequencing, because for short read, although we have a reference genome, but there's still a lot of gaps when you try to identify variants, and also especially for large structural variants, short read won't be good at it. Okay. And then this is the true advantage that comes from long read and sequencing. And also, you can, in a way, for example, if you use PacBio Revio hi-fi read, and then you can really accurately call small index and also SNVs as well. But you can also get much better variant calling for large structural variants as well, and potentially can do phasing on your SNVs as well. So which cannot be really achieved by short read. Depends what would you want to achieve through resequencing.
Perfect. Thank you, Dr. Shichi. So thanks for everyone's questions. Unfortunately, that's all the questions we have time for, but don't worry. As mentioned, if we haven't managed to answer your question right now live, we will get back to you via email by next week with a full answer to your question. And so thank you, all for your time today. We hope that you enjoyed this webinar. We hope you found it useful. We will email you a link to the webinar recording as well as the slide deck, so you can expect to receive this on Friday. And when the webinar finishes, a survey is going to pop up. We would really appreciate you taking the time to complete this, because it helps ensure we make future webinars even more relevant to you and your research goals. So there will be a link in the follow-up email for you to view a certificate of attendance as well, should you need one. And this can be just printed from your browser window. In the meantime, you can follow us on Twitter and LinkedIn, and there you can keep up to date with future webinars, offers, and news from us. So thanks again, everyone. Thank you, Dr. Shichi, and we look forward to seeing you all on the next webinar.